Okay, so yeah, right now Claire is sharing our master document, and this is the one where we have, as you can see, the first link there is our ongoing user group notes where we keep, I guess, all our, pretty much what you were just on, what you're putting your attendance on. And then we'll have each week will be each month we'll be populating it with the notes that we take during the meetings. And then the list that Claire is, or the document that Claire is showing right now is our master document. And that's where we want to keep all the links and all the references or resources that we want to provide you guys with. So I guess I spoke about a little bit about it in the emails, but I've created some Google submission forms where in previous meetings we've talked about wanting to share our experiences with different fluorescent proteins, fluorescent dyes, or reagents that people have used for expansion, whether things work or they don't. So you're welcome to click on each of those links and fill in the form. You can fill it to the best of your abilities. If some things are missing, that's okay. We just kind of want to get a bigger picture about, what people have done or have used and what works or doesn't, so that, we're all not wasting a lot of resources and time trying reagents that, everyone else has already tried and has shown that doesn't really work very well with expansion. As well, you can see that there's two other submission forms that would be the speaker suggestions or sign up and the meeting topic suggestions. So for the speaker suggestions or sign up, we're pretty much leaving it open. You can submit somebody, or you can suggest somebody that you want to hear from, and we will work our best to invite them and have them speak on the topic that you would like to hear from them. As well, if you have something that you want to talk about or present, you're also welcome to sign up and volunteer to present on your own. We're also very happy and open to that. As well, the meeting topic suggestions would just, so you don't have to present on that topic, but it's just what you would like to hear or what you would like to learn more about in future meetings. So yeah, does anyone have any questions, or can everyone access them? You can try now and let me know if, if it doesn't work for anyone. As well, for these forms, we will hope to be generating like an Excel sheet from them eventually to kind of compile everything to make that accessible to everyone. And yeah, that's all. And feel free to let me know if when you're filling out these forms, you feel like there's like a question that you want to ask, but I don't have that question there, or if you have any feedback on the form, you're also welcome to put that either on the form or just shoot me an email and I can add those for you. So yeah, right before we have our presenter speak today, also one more thing about our upcoming meeting. So we have decided, we've Gabriella from the university in Australia, she has decided that she will be able to host like the parallel meetings for us. But we think that because we're recording these meetings for them and they will also be recording their meetings and speakers for us, we can probably have our meetings every 2 months instead, so that we will be on the same page and we'll always have some kind of speaker or material to talk about. So yeah, our meetings probably will take place every 2 months from now on. And then in the month that we're not having a meeting, it'll be the Australian hosted having theirs. So you're welcome to attend both. They'll both be recorded and uploaded for everyone to access. It's just you won't have the live session or the discussion parts available. And yeah, so without further ado, we have our guest speaker. She's coming to us from Italy. And I don't know if Chantal, you want to introduce yourself and give us a little background?
Yes. Hi, everyone. I am Chantal Usai, and I am a PhD student in the Italian Institute of Technology in Genova, and I work under the supervision of Professor Alberto Diaspro and Professor Paolo Bianchini. My background is in chemistry, and now I'm doing my PhD in biophysics. and I started working last year with expansion microscopy. So this is my background. Okay. Okay, should I start sharing my screen? Okay.
Thank you.
Okay. Okay, can you see my screen?
I think we see your split screen. Okay, yeah, that's good.
Okay.
Perfect.
Okay. Okay. Okay, so let's today we're gonna talk about the problem of expansion validation, and especially how this problem has been faced in our group using the nuclear pore complex as an intrinsic reporter. So, as we all know, even though expansion microscopy is quite recent technique, the panorama is already quite vast, and we have a lot of protocols already available. But all of these protocols start from the same point, which is the synthesis of a soluble polymer network inside your biological sample. Then can then be physically expanded, and this results in a physical magnification of the sample. So we all know that, but when you work with expansion microscopy, it's very important to see each step, to see this system as an engineerable system, and you can customize each step in order to achieve what you want to achieve. So starting from the labeling, we know that in the original expansion, the first protocol from 2015, they were using a three-functional fluorescent label featuring oligonucleotides sequence to target the primary antibodies for the immunostaining in the sample. Then, starting from that, the protein retention expansion microscopy protocol was developed, and so this allowed for the use of standard conventional antibodies and introduced for the first time the concept of post-expansion staining. And then after that, we also know that a lot of labs around the world are now developing gel anchorable small molecular probes for various biological targets. Then, speaking about the second step, which is the gelation, where you synthesize your hydrogel-based polymer, we know that most of the polymers are based on these two monomers, sodium acrylate and acrylamide, that you can see here, and then usually there is a cross-linker to regulate the expansion. But now we also know that modifying the composition of the gel and the relative amount of these monomers can have an effect on the preservation of different biological structures. Then we have the digestion step that originally started as an enzymatic digestion using a proteinase, but then in different protocols during these developments, we discovered that modifying this digestion buffer, you can target it to your specific biological sample. And for example, you can supplement it with specifically enzymes to digest your sample. But in some cases, you can also completely replace this enzymatic digestion with the denaturation step, which can be either thermical or chemical. And then, of course, we have the expansion by water dialysis, and then we can finally image our sample, obtaining super resolution imaging thanks to the physical magnification of our sample using a diffraction limited microscope. So we're starting from our cell, for example, from our biological sample. In our case, let's consider a cell, and we have to always keep in mind that there is a heavy sample processing involved. And although for some application, like for example, the mapping of some neuronal network, the quantitative retention of the ultrastructure is not really needed. If we are talking about cell biology, the necessity for the validation of the ultrastructure retention becomes very important. And also you may need to be able to quantify your expansion factor in a more accurate way than simply measuring your gel size at the end of your expansion. And so in the last few years, a few examples on how you can validate your expansion protocol basing on reference structure came out. Let's step briefly back and see how the validation was performed in the original protocol. As we know, the first validation was a comparative analysis between pre-expansion imaging taken using super resolution microscopy and post-expansion confocal images. In the first paper of Professor Boyd, and they used structural illumination microscopy as a super resolution microscopy technique, and post-expansion they used the spinning disc confocal. They use as a reference structure microtubules and cluttering coated pits, and they perform a measurement error analysis, which gave a root mean square in the order of 1%, which is well within the point per expansion of the super resolution microscopy technique that they used. So we can always perform this comparative analysis to validate our expansion protocol. So for example, we can take a large image pre-expansion and then post-expansion, we can go find back the same cells, and then we can obtain something that looks like this, which is always valid. But so you first need to do a big mapping on your sample, and then you need to find back the same cells, which is not always simple. So this is a very time-consuming method. That's why we can try to use some intrinsic reporter as a method of validation. So to be able to use a biological structure as a reference, we need to know this structure very well. So in our lab, we started using as a reference structure the nuclear pore complex. The nuclear pore complex is responsible for the transport of material between the nucleus and the cytoplasm. And most importantly, for what we're interested in, is a very well characterized and known structure. We know that it has an octagonal symmetry around the axis of transport, and that it is pseudoplanar through the nuclear envelopment. Then we know the diameter of the structure, and several of the nucleoporins that compose this structure have been characterized in super resolution microscopy, and the primary antibodies to target these nucleoporins are commercially available. So in our lab, we decided to use a reference structure, the nucleoporin 153, using a primary antibody produced by ABCAM. So to validate for the first time this validation method, what was done at the beginning was to simply perform a confocal imaging before and after the expansion on the same cells. Then this image can be overlapped and rescaled to a fine registration in order to perform the same comparative analysis that was performed in the past to validate expansion microscopy. Then using the pixel size ratio between the images, we are able to measure the expansion factor on a nano, micro, and macro scale level. But what we want to do is to perform a statistical analysis in order to remove the need to do this comparative analysis before and after the expansion. So what we did was to analyze the nuclear pore complex at different expansion factor and with different optical resolution. So we performed an expansion STED experiment And this needed, there were a lot of parameters that needed to be finally tuned experimentally. And this analysis was performed at 3 different expansion levels. Before the expansion, so on our biological sample as it was, post-digestion, since after the digestion you usually achieve a first expansion with, which in our case has an expansion factor around 2, and then at the end of the full expansion in water, which in our case has a macro expansion factor around 5. And here you can see the line profiles of the selected nuclear pore complex unit compared at the 2 different optical resolution and in the 3 different expansion levels. The next step was to start this determination of the expansion factor at the nanoscale in a statistical way. And this was done using image analysis with a custom MATLAB algorithm. So what was done was to first measure the radial intensity profile of a single nuclear pore complex unit along different orientation. And in doing this analysis, the algorithm is able to calculate the radius of the pore and the position of the center. So we can have averaging all of these profile along different orientation, the final radius for each pore. Then this statistical analysis was performed on a large number of nuclear pore units, and then we can plot the sum, the distribution of all of these collected radii and fit it with the Gaussian distribution for the 3 different expansion levels. And then we can obtain the nanoscale expansion factor that we can compare with the macro scale one that we had by measuring our gel. Then we wanted to determine if the expansion was isotropy or wasn't. And this was done again using image analysis. So for each pore, we know that the maximum value of the radial intensity can be plotted as a function of the orientation. So what we did was to fit with a multi-Gaussian peak this function, and we used the first subunit as a reference, giving it so attributing the angle 0. So based on the statistical nature of the immunostaining protocol, we know that in most of the pore we will not have all of the different subunits immunolabeled. So this analysis was performed again on a large number of nuclear pore complex units, obtaining then all of the subunits in the cumulated data. So we can plot the final accumulation as a histogram, and then we can retrieve the angulus values, which we can fit with a 7-peak Gaussian. And this is used to demonstrate the isotropy of the functions, since the angular difference among these units is a multiple of 45 of the angle of 45 degrees. The last thing we did was to try to estimate the resolution of our system. So first, we wanted to determine the optical resolution of solely the STAD imaging. So to do this, in order to do this, we perform STAD images on non-expanded sample using the same imaging setting that were used for the 3 experiments: pre-expansion, post-digestion, and post-expansion. So then from the depletion curve, we were able to extract the K value in order to calculate the improvement in resolution, considering a confocal resolution of 250 nanometers. And then to calculate the effective resolution of our STAD expansion experiment, we normalized the single optical resolution for the 3 conditions by the expansion factor for each condition. achieving the effective resolution that you can see here in this slide. A similar work has been done in the lab of Professor Paul Guichard using centriole as a reference structure. So in this case, the comparative analysis was based on the ninefold symmetry of isolated center, and they used it to perform imaging, direct stochastic optical reconstruction microscopy, and scene microscopy. This a very interesting point of this work is that it shows how slightly changing the gel composition and the fixation method can have results on the different preservation of the biological ultrastructure. Because in this work, they compare the original expansion microscopy protocol with the magnified analysis of protein 1 and the ultra expansion microscopy method that they develop specifically to better preserve the ultrastructure of centrioles. Another way to quantify the expansion factor at the microscale level and to determine if there is a distortion during the expansion has been using this intrinsic in this very interesting work using fluorescent photobleaching. So what they did was to transfect HeLa cells with a plasmid to induce the expression of nuclear enhanced green fluorescent protein. And then they used two-photon excitation to induce photobleaching of the EAGFP, introducing well-defined 3D features in the cells. So they basically bleached out some cubes inside the nucleus of their cell. And then they used these cubes as an intrinsic ruler to validate the expansion factor and the sample distortion in their samples after the expansion, measuring the dimension of this cube after the expansion. And what they found is that in their experiment, the expansion was isotropic in all 3 dimensions. Then, before introducing the last expansion factor validation method that I would like to try, I will briefly tell you what I work on now during my PhD. So now I'm working on the investigation of modification in chromatin compaction related to genetic diseases, and I use expansion microscopy combined with other super resolution microscopy methods. Particularly, I'm working on Hutchinson-Gilford progeria syndrome, which is a genetic disease which causes the early onset of aging symptoms. And this disease is caused by a single nucleotide mutation in the lamin A gene that causes the production of the modified version of the lamin A protein called delta L50 or progerin. The presence of this protein modifies the dynamic and the structural protein, the structural properties of the nuclear lamina, especially leading to nuclear complex clustering, the thickening of nuclear lamina, and the loss of peripheral heterochromatin. Our cellular model is based on human epithelial kidney cells, and we have 2 groups of cells in order to perform a comparative analysis. In the first group, we induce the expression of the modified protein delta L50, while in the second one, we examine the wild type form of the lamin A. Both are tagged with this C3GFP, which is a modified version of the GFP protein that has been shown in the past to be efficient for cell microscopy. And to counterstain the DNA, we are using the SPI 650 DNA dye, which is a far-red minor groove binding DNA binding dye that gives very good results with the post-expansion staining as well. So it's a very interesting dye for expansion. So what we have seen is that in the HDPS sample set, there is a large variation in chromatin compaction, and this seems to affect the physical association between lamin and heterochromatin in the nuclear periphery. And seems to lead the proper spatial separation. So we wanted to quantify this distance between these two structures, and we are doing this by analyzing the intensity profiles of these two of the two structures to determine this distance between the DNA and the lamin. So our preliminary results show that the two groups seems to belong to two different populations. But since we are using expansion, we would like to further investigate if since the two structures that we are examining, and one is based on DNA and the other is a protein, we would like to investigate the different behavior of these two structures during the expansion. So we would like to try a microscale expansion factor validation based on DNA. So we found this very interesting work that is being carried on in the Gatta Quant Lab, where they use the DNA nanorulers, which are the DNA origami-based nanorulers that have two fluorophore at a very well-known distance. They are usually used to quantify the resolution of an imaging system. Basically, they use the modified version of the nanorulers covalently bonded with an acridized molecule. So these nanorulers can take part in the polymerization process during the gelling, and then after the denaturation step can be expanded. So what they did in this work is that first they quantify the macroscopic expansion factor, as we all do, and then they perform a distance analysis on a very large number of DNA origami, obtaining the macroscopic expansion factor. The interesting part of their work is that they found that there is a deviation between the microscopic expansion factor and the macroscopic one. The microscopic seems to be slightly smaller. So what we would like to do is to implement these nanoruler tools inside our biological sample to be able to perform 3D studies and quantify the expansion of DNA inside our sample. So, in conclusion, we can say that in the last few years, the intrinsic reporter has been successfully used for rapid expansion microscopy validation. Although this is a process that needs careful optimization, especially if you use less robust biological structure. But you always have to keep in mind that if you have deformations on your sample, the tuning of the gel receipt you're using can mitigate this effect. And also, you can always have the possibility to add external reporters to your gel. But in conclusion, we can say that it doesn't exist a universal method for retaining all of the ultrastructures in your sample yet. So there is still a lot of work to be done. I would like to thank all of the people I work with every day, and I would like to thank you for your attention. And if you have any questions, I'm open for discussion.
I think Mattia has a question. You can unmute yourself.
Hi, thanks for your presentation. So I have like a biological question about the experiment that you're trying to do with the DNA. So
Okay.
Why didn't you use a heterochromatic mark in your experiment? So why you didn't use maybe a mark for, I don't know, like lysine modification that you can tag with and you just get the heterochromatin?
I actually did that. Like here, I just show you the one with the full DNA. There is not I performed the image analysis only on these ones for now, but I also stained the methylation modification on the lignin 9 of the histone tree. So well, I didn't want to talk about my work too much because this wasn't the point of this meeting, but we would like also to study the modification in both heterochromatin and euchromatin in this biological sample that we are using now.
Okay, thanks.
You're welcome.
So
go ahead, Natalie.
No, I was just going to read out what people mentioned in the chat. Javier said, excellent presentation and results. Congratulations. He wanted to thank you, but he had to leave because his teaching duties called him back. But so that was the first comment. And then Chris Chan asked, how vary every time do you make like an expansion gel or the range?
So I assume you mean like the expansion range, like if the expansion is the same? Is that the question? Okay, the expansion factor. Now I am using mostly the protein retention expansion microscopy protocol. So using the same condition, and I use the same also the same monomer solution because I made an amount and then I freeze them in aliquots. The expansion is usually around the same value. So every time I measure the gel, and usually the expansion factor I have now is usually around 4, and usually is quite consistent. You're welcome.
Claire, did you want to go ahead?
Yeah, I had a question about the DNA origami. Just how easy is it to get it evenly distributed throughout the gel?
Okay, so this is something that we are just starting to do. We bought the DNA nanorulers with this acridite modification already, and we didn't start yet to add it into our sample. So this is something we want to do, like we are ready to do now. But in this work, they seem to be very well distributed because they put it on the cover slip, so and then they synthesize the gel, and I think we will do the same thing because we have the cover slip with ourselves. So then we will infuse with the DNA nanoruler solution, and then we will add the monomer solution. So that's it.
Keep me posted. Thank you.
George, do you want to go ahead?
Yes, excellent presentation. That was a very nice summary of what's been done so far and a great introduction to the topic as well. I had kind of 2 different questions. One, for the nuclear pore complex labeling, are you using a primary antibody and secondary antibody complex?
Yes, at the moment, yes.
Have you considered using a conjugated primary or a nanobody if that existed, or doing post-gel staining, which has started to become popular in the literature?
Yes, we would like to do the post-expansion one, the post-expansion staining. We would like to try. I didn't try with the nuclear pore complex yet. And also we have the nanobodies for the secondary antibodies, and this is something also we're starting to use.
Okay, great. My second question is for protein and nucleic acid anchoring into the gel. I know the early protocols had kind of very specialized anchor preparation rather than the original Acra Oil X. But I know there's been 2 recent papers that have shown alternative anchors that work with both protein and nucleic acid intrinsically. Are you doing the kind of 2 anchoring step, or do you have a combination anchor?
At the moment, we are using the ACX anchoring, so it's the one based on the succinimide ester, and we didn't try. Well, we tried the first one, the MAHS, but now we move to the second one, which is the one that they use in the protein retention expansion microscopy, which seems to be better good results for us. But we will also try to do something else. I found an interesting work they used to combine expansion microscopy with Raman, so this is a different kind of work, but they seem to have a very high protein retention there, so this is something I would like to try. I don't know if I answered your question.
Yeah, that's very helpful. Yep, thank you very much.
All right, so Michael Cameron actually had a question, but for some reason his hand raising option wasn't available. So Michael, do you want to proceed?
Yes, thank you very much. The validation work is very important. I really enjoyed your overview in this talk. But each validation experiment uses a single molecular structure as a reference. So how do we know that all structures expand the same amount, such as centrioles versus potassium channels or microtubules, or maybe different cell types in different tissues? So it's both a practical question, but also sort of a philosophical question, like, does it really matter for most work, because we're not really comparing different structures? We might only be looking at what's going on in each structure at a time. Thank you.
Yeah, this is something that I think about too. In some of these work, they use 2. So, for example, I didn't show it, but in the one they did in my lab, they also stain the cytoskeleton, and also in the one in the ultra expansion microscopy protocol, they also stain. They use again the cluttering pit to observe another reference structure. But yeah, that's a very good question, and that's why at the end, my point is that there is not a universal method, because every work only looks usually at a few structures at best, because you cannot examine everything in your sample, of course. But that's a very good question. I do not have an answer for that, but it's a very good question. There's something to think about.
All right, and you have a question in the chat. Have you seen a difference in the loss of signal after expansion between the different secondary antibodies, and which one do you use?
Yes. Other than the one that don't work, like for example, we know that some don't work at all. For example, the Alexa 647 doesn't work for expansion. All of the secondary antibodies I work with, I found that the Alexa 488 is the best for expansion, I think, at least for pre-expansion staining. Unfortunately, since I'm using GFP, I cannot use it. but yeah, and then I tried the others. For example, the up to 647N for me has quite good retention. Also, the Atto 594 is another antibodies that I'm using, and I also tried the 532, but I think the retention is a little less. I didn't show it here, but something that I started doing some time ago is to try to restain the secondary antibodies after the expansion. And usually this, I don't have the image in this presentation, but the signal to noise ratio gets a lot better, assuming your primary antibodies were not digested by the digestion buffer. Like if the primary survives, then the secondary staining can be a solution.
Okay, perfect. And one more question from Julie. She asked, what kind of mounting medium do you use for said acquisition on your gel?
Well, the gel is just enclosed between two cover slips, and then is sealed with a big component polymer, but like it's just to seal the gel to prevent the shrinking due to evaporation. But between the cover slip and the gel itself, there is nothing, like you imaging the gel on the cover slip. So that's what we do. And for the let's say not final fully expanded gel, we just do the imaging on the cover slip. For example, pre-expansion, since afterwards we want to expand it, we just image on the cover slip mounted in a metal ring with PBS on top. Welcome.
Chantal, do you think we could ask you to put your experience with the different antibodies into the fluorophore form, just so that we have a record of the ones you've found work well, and also put in the ones you found don't work well, so we can keep.
I will definitely do that.
Thank you.
Thank you. Okay, I'll stop sharing my screen.
All right, thank you so much. That was an amazing presentation, and I'm sure we've all gained a lot from that. And I really like the part where you talked about other people's research and what they're working on and how that can kind of be incorporated into your work, and just you really focusing on the validation that's happening everywhere. That was really helpful, is what I found. So thank you so much for that. We will record that and have that posted. So I don't know, maybe people from the Australian meeting will also email you if you're okay with that, if they have any questions. Okay, sure. Perfect.
Thank you.
Easy. So I think, I guess one more thing that I forgot to talk about earlier today was the RMS registration. So since you are all here, I assume that you have all registered for the mailing list and are all able or receive the invitation, whether you've registered. So there's two options. There was one where you can register just for the single meeting or for the series of meetings. You're welcome to do both, but you do have to make sure that, if you've there were two registration forms. One was just for the RMS mailing list, and the other was specifically for our event. So yeah, make sure you do the one for the event, but you do have to sign up for, I think, or input some of your personal information in order to get into the event, right, Claire?
Yeah, so I guess it's one form, but you need to select if you want to just be on the mailing list or if you also want to sign up for the actual individual meetings.
Mm-hmm. Yeah, and you're welcome to share that with anyone. Like, everyone is available, like, as long as they're willing to register. We're open to having anyone join our group. Yeah, so does anyone have any questions or any comments? Okay. If not, I think, yeah.
I was gonna say, I think we'll keep our meetings at 1.5 hours just so we have that flexibility. And I think everybody's always happy to gain a half an hour back in their calendar. But our next one will be on Wednesday, December 7th from 9:30 to 11 Eastern time. And we will send out information about the Australian time zone meeting, which will be in the first week of
November.
Sorry. Yeah. Thanks, everybody.
Thank you.